Good evening or morning. Well, I don't know, whenever you're watching this. This is a JS110 Introduction to Forensic Science. I'm Jason Linville, and this is the lecture on identification of body fluids. And we are recording this in the fall of 2017. So anything about the course material obviously still applies, but if I mention anything about quizzes, exams, assignments, uh, anything else that's going on in September, it's hurricane. It's all hurricanes all the time in September of 2017 and the Cleveland Indians. You can ignore all of that stuff and just focus on the material. All right. I begged, literally, right before we began, somebody to come up with a 2 to 3 sentence story that involved body fluids, and either there's a delay or nobody took me up. I should have pointed somebody out, right? But this is how we like to begin a lot of these lectures, probably regardless of what semester you're in. We started off with some education and CSI stuff, and now we're into different types of evidence that come into the crime lab and going through the procedures of what the lab will actually do. So this second one, body fluids, we're actually going to cover a variety of body fluids here, mainly semen and saliva. I mean, really blood and semen are the most common pieces of evidence, biological evidence that come through a lab. And then we'll also just mention if you can do anything with other body fluids. All right, so a quick case would be, let's go ahead and go to the next slide here and make sure my clicker is working. So a brief, oh good, see I talked long enough. Sarah stepped up to the plate and describes a case here. Okay, so we have a female was sexually assaulted in her apartment. After a sexual assault kit was done, it was determined that the attacker left behind semen in the victim. Semen was also found on the female's underwear. Okay, so there we have a swab from a sexual assault kit, which we're going to talk about in this lecture. And we also have a pair of clothing, fabrics that might have a biological stain on them. Okay, now, so let me go ahead and draw the swab from the sexual assault kit here, okay? All right, now, John Thomas has come in with another one where, let's see if it's similar. Taylor Swift had a boyfriend who she thought belonged with her. Well, this could be applied to many different types. They made love, he killed her, and now there's bad blood on the floor. No, that's blood. That was last week. Although I'll give you extra points for incorporating Taylor Swift and several names of her songs and reputation in there. Okay, I'm going to stop talking about Taylor Swift because I was already talking about Taylor Swift last week and in another class this week, and it's really it's a little bit too much for me. Let's go back to this wonderful drawing I have of a sexual assault swab. And the first thing that you want to know, especially with the sexual assault swab, is semen present on that? And to do that, it's this swab. Okay, go ahead and answer in the chat box or shout it out at home. Would this swab be our known sample or our unknown sample? And again, this is some preparation for mock quiz questions, which I think we have a mock quiz coming up next week that we want to be prepared for. So the first thing you need to do is identify this as a known or unknown sample. All right, and Sarah comes in first and is correct. Anytime we have evidence, it is an unknown sample. Okay, so now we need to ask ourselves, okay, what is the known sample that we're going to compare it to? So again, you can throw this in the chat box or you can answer at home. We need to identify a known sample to compare it to. And when we're dealing with identification of semen, it's going to be very similar to the identification of blood. And we talked about how if you want to know who it came from, then you're going to have to get, that's really DNA, and you need to get DNA from your suspect. But here, and we have a correct answer in the chat box, our known sample is actually going to be something that we know is semen, okay? And this becomes not necessarily a comparison where we're trying to identify the origin of the semen, because we'll get to that with DNA. But here it's an identification of a substance. So just like with blood, we had presumptive and confirmatory tests. Just like with drugs, where we're trying to figure out what drug is this, not where it came from, we have presumptive and confirmatory tests. Here, with semen and saliva and the others, we're going to have presumptive and confirmatory tests. Okay? Now, in order to identify good presumptive and confirmatory tests, we need to start thinking about the traits of semen. So what are some traits of semen where we can compare the unknown sample to the semen to see if they match? Think about, and again, shout this out at home or throw it into the chat box. Think about what we did with blood. Yeah, we talked about the color, and the color of blood is reddish. With semen, it's sort of whitish or clear. What were some things in blood? Well, let me rephrase that. We talked about things in blood, like the cells or the proteins. Can anybody think of any cells that would be in semen or proteins that would be in semen? Okay. And when it comes to cells, we're going to be talking about sperm cells. And this is actually going to be very good because sperm cells are a little tougher than blood cells. They don't break open, they don't lyse as easily. Unlike with blood, where we couldn't look at a blood stain and see the blood cells under a microscope, here with semen, we're actually going to be able to find these sperm cells. Okay. And then, well, there's proteins and stuff. Hey, again, Dr. Linville, this is why I'm in the class. I don't have all of the proteins in semen memorized, and I don't expect you to, but two prominent ones are going to be acid phosphatase and prostate-specific antigen. So that's the whole purpose of this lecture. But actually, just like blood, we're going to do chemical tests to detect the presence of those materials, along with an antigen-antibody reaction. Okay? So again, when it comes to all of these traits, what we need to ask ourselves are, okay, if the trait matches, in other words, if we if we look at that swab and we see that it looks like it has a whitish to clear liquid on it, are there other things that are also whitish and clear? Yeah, it could even be like vaginal secretions or something like that. So that's not a very specific test. Okay, what about sperm cells? Are there things other than semen that have sperm cells in them? Well, as it turns out, there's not. Are there other things that have prostate-specific antigen in it? Well, it turns out it depends on the tests you run. And all of these things, again, unlike the pencil, where we sort of know enough about pencils to answer questions about color and logo and stuff, that's the whole point of this lecture. We want to look at these traits and find out, are they specific to semen, meaning it's more of a confirmatory test, or might there be some false positives involved? Okay. Now, I try to emphasize this a little bit more in later classes, like if you take JS 352. But it's important to know that with blood, if you do a TMB test and you get a positive where it's presumptive test positive for blood, and you do DNA testing, that you don't really go into court and anybody questions whether it was blood. Okay, it has human DNA in it, and it looks like it has hemoglobin in it, and it was found, next to a bleeding body. It's probably blood. So the question of whether it is blood doesn't become that important to an investigation. Okay, with semen, on the other hand, many times in an investigation, you want to go into court and be able to say with confidence and confirm that yes, this was definitely semen. Okay, because if you get DNA out of it, then the defendant may say, oh, well, yeah, I was present in the room with the victim, but it was saliva. We were just making out, but it's not semen. Okay, so it's much more important from the forensic scientist perspective to be able to go into court and say, yes, this was definitely semen. So with blood, where confirmatory tests aren't necessarily done all the time, or in some cases at all, with semen, we're going to find that the confirmatory tests are much more important. Okay, not only can it link us to the attacker, down here, identify the perpetrator, but just being able to say that it's semen can help provide further proof that a sexual assault did take place and, that it was a crime. All right, so let's start looking at the traits of semen. If you do a search for semen on the internet, safe search on, then you may get some things like, oh, it's sperm cells, and there's some more sperm cells, and wait a minute, something's not right here. This came up, and I threw it in this lecture, I don't know why, but it turns out what's going on here is they're artificially inseminating this elephant. So this is actually an elephant here with the tail hung out. And that, sure, semen, artificial insemination. But what I was curious about is, number one, why is this woman smiling so much? Number two, this guy even tail guy looks like he's smiling. And number three, most importantly, is we got 2 feet down there, and I'd really like to see the reverse angle to find out what in the heck that Blue Apron guy is doing. I usually regret talking about artificially inseminating elephants at this point, so instead I'll show you a picture of a kitten, and then we'll just move on to the next slide. Okay. All right, so what are some traits of semen? We said that it's clear whitish, what's in semen, cells, and enzymes, and that's what we're going to talk about. Now, it turns out sperm cells, as it says here, are the most important component, and most important in a forensic context is kind of a weird term to use, but the reason why you would prefer to find sperm cells in your evidence sample is, number one, as it says there, it's specific to semen. So are sperm cells in any other material? No, they're not. So if you find sperm cells in your sample, you can say, yes, this is definitely semen. Okay? It's a specific trait. The other reason sperm cells are important is it's kind of easy to describe that to the jury, right? That, if you start talking about PSA tests and antigen antibody reaction, the jury might be able to get a little confused. But with sperm cells, it's like, oh, okay, they found sperm cells. That means semen's present. Okay, sperm cells aren't in saliva or blood or anything else. Okay. So unlike blood, if you have you can have a small cut, a large cut, you can bleed a little, you can bleed a lot. With semen, sort of the evidence from one incident is a controlled volume, so we're talking 2 to 6 milliliters with 100 to 150 sperm cells per milliliter. So if you do the math there, which is not terribly complicated, 600 million sperm cells, there is plenty of sperm cells in a sample to do DNA analysis. So usually doing DNA analysis, even if it's where you're recovering it from an orifice with a sexual assault kit, or if it's just recovered from clothing. As some of our research at UAB finds, even laundered clothing may still have plenty of sperm cells on it. The sperm cells tend to be tough to get out of fabric. So there's going to be plenty of sperm cells for, number one, the test where we look for sperm cells. We'll talk about that later. And also for DNA analysis. However, the sperm count may be lower or zero due to disease, genetics, drugs, alcohol, or a vasectomy. For those of you not familiar with a vasectomy, I'm going to draw a slightly graphic picture here. Oh, and snip. Okay, so these are scissors here, and then this is the testicle down here, and this is the business end. And what happens is sperm cells are produced in the testicle, and then they come out the business end during I don't know what, sexual intercourse. I guess I should use the biological term. And if that tube is snipped with scissors during an operation, then that's a vasectomy, and you're not going to have any sperm cells in the semen. Okay, why do men get vasectomies? I always like to tell the story that my sister and brother-in-law flew from Minnesota to Washington State with 2 young children, and they decided that they did not want a third. So when that happens, if the male gets a vasectomy, then in theory, unless it magically heals itself, not magically, unless it biologically heals itself, which is rare, then there you go. So you can sit down and start writing your crime novel about the rapist with a vasectomy, but it's important for forensic scientists to know that if it looks like semen is present due to all of the other tests, but they can't find sperm cells, there's a possibility that you're dealing with an individual who has a very low sperm count or a vasectomy. Okay, this is what sperm cells look like. So they look like tadpoles. And we do refer to the parts of the sperm cells as the head and the tail. The head is where the DNA is, and if you've seen the movie Look Who's Talking, congratulations. If you have seen the movie Look Who's Talking 2, then my condolences. But at the beginning of the movie Look Who's Talking, where John Travolta and Christy Alley have a baby who's voiced by Bruce Willis, the sperm cell is also voiced by Bruce Willis, and there's sort of this neat movie, like biology class movie of the sperm cell, penetrating the egg cell. But the head of the sperm cells is what goes in, combines with the nucleus of the egg, and then boom, there you go, you got a human. The tail is for movement, so there's not really any DNA or anything in the tail. And also, I think the book just adds this in to confuse you. The acrosome is a structure at the front of the head of the sperm cell that does contain enzymes, but what I'd like to emphasize here is the enzymes in semen that we're going to talk about are not in the sperm cells, not associated with the sperm cells. In other words, if a person has a vasectomy and 0 sperm cells, it's not going to affect the other tests that we're going to talk about, okay? The presence or absence of sperm cells will only affect the test for sperm cells. Okay, number 1, searching for semen stains. You look for them just like you do with blood, and if blood's on a light fabric, it looks a dark brown color. It's pretty easy to identify. Wait, I haven't been paying attention. Keith, have you had a vasectomy? Wait, is this a personal question? Or no, wait. Keith has not had a vasectomy. He has seen the movie Look Who's Talking. Okay. See, that's a problem. If I don't pay attention to the chat box, and all of a sudden I've accused Keith of having too many children and not wanting any more. Okay. So it would have been weird if, like, Sarah would have. I don't think I would have made that same mistake. So semen stains appear yellowish white, but just like if a blood stains on dark fabric, semen stains can be difficult to see on dark fabric or light fabric or on a swab from a sexual assault kit. What you can do, especially if you're searching clothing or bedding material for semen stains, you can use an alternative light source or an alternate light source. You can use UV light, which is what we talk about here. You can also use blue light with orange goggles. If you've seen any episodes of Hotel Nightmares, that's what they're doing there. But the important thing to understand is that semen does have substances in it naturally that will cause it to fluoresce. So you don't have to apply any chemicals like you do with blood. Semen will fluoresce naturally. Now, there are a lot of other things that will fluoresce. So, for example, if you're watching a television show where they go into a hotel room, get out the black light, and it looks like there's semen stains all over the place. Not necessarily all of those are semen stains, but it's still pretty gross. Hey, wash the comforter every once in a while, right? Okay. If you're searching for semen stains on a blue dress, for example, you've time traveled back to 1998, then yeah, you would do the same thing. You would go into the dark room, look with your UV light, and semen will glow. Then usually what you do is you take a Sharpie, you put a circle around the glowing area, and then you take it out of the dark room into the lab and perform some of these tests that we're going to talk about. All right, a little heads up. We're going to talk about one presumptive test for semen. So this is just like the TMB test, the presumptive color change test that we talked about for blood. And then we're going to talk about 2 confirmatory tests for semen that are a little bit new. Okay, the presumptive test is for seminal acid phosphatase, SAP. One of the confirmatory tests is going to be for prostate-specific antigen, PSA, same 3 letters, different order. So start focusing now on differentiating these 2. Instead of saying SAP, say acid phosphatase. Okay, that's how they refer to it in forensic science labs. Your book calls it the Menemine Fast Blue test. I don't know what nerds call it that or if it's a regional thing, but I've only heard it referred to as the AP spot test, which is a test for acid phosphatase, specifically seminal acid phosphatase. Now, the test will react with other acid phosphatases, so it's important to understand what type of materials other acid phosphatases are going to be in. Urine, vaginal secretions, perspiration, cauliflower stem juice. One of those 4, and go ahead and take a guess at home or in the chat box, one of those 4 is going to be very important for forensic scientists to understand as a false positive because it's going to be mixed with semen in a lot of samples. Can anybody identify? I don't know. This might be easy or it might be difficult. Can anybody take a guess there? Yeah, you might think, oh, well, maybe urine because of the man's urinating and what. No, it's vaginal secretion because, again, in a sexual assault kit, if it's a vaginal swab, then you're going to have vaginal secretions on the swab no matter what. So you're always going to get a slight positive to this test. But the fact that seminal acid phosphatase is in such a high amount and is very reactive in the test that we're going to describe means you get a much stronger reaction when semen is present. So again, all this boils down to is the experience of investigator. If you get a strong reaction, you know it's semen. If it's a weak reaction, then you have to use your best judgment about whether, to move on and test for semen or whether you think it's just, vaginal secretions or some other substance causing a weak reaction. Okay, So the spot test that we're going to talk about will react with dilute semen or even semen in small amounts. The acid phosphatase, seminal acid phosphatase, is high in males until age 40 and then gradually decreases, but it's still in detectable amounts. Again, if you want to write a crime novel about a 90-year-old man with a vasectomy, then who is also a rapist, I guess we should throw, and time travel. No, just rapist, no time traveling. Then that would be challenging for a forensic scientist. The SAP comes from the prostate gland, so again, it has nothing to do with sperm cells. Okay, if a person has a vasectomy, then provided that they're under age 40, or they're still going to have a large amount of acid phosphatase in their semen. Okay, what is the reaction here? Okay, remember when we were testing for hemoglobin, okay, we added two things, a chromogen and an oxidant, and they reacted, and in the presence of hemoglobin, they reacted faster. Okay, here we have seminal acid phosphatase. That's what we're going to look for in semen, so this sort of plays a role with the hemoglobin. And we still have a chromogen, and for you guys, I don't think you need to, I use this slide in another class as well, so don't really worry about the name of the chromogen, because just know that this is the AP spot test. Okay, and there's also a substrate. So what happens, if you're curious, is the semen, that seminal acid phosphatase, reacts with this naphthol phosphate, cleaves the phosphate, and then the product reacts with the chromogen and causes the color change. Okay, what you need to know is in the presence of these materials, which are mixed together in a reagent, so it's not like the blood test where you add them separately. You mix these together, you add 1 drop on your sample, and if it turns purple, then that means it's could be semen. Okay, if it doesn't turn purple, then there's probably not a lot of semen there. Okay, how do you actually do this test? Okay, how you do this is if you're looking at a semen stain on fabric, then just like the blood test, you would take a swab, moisten it with water, rub it on the semen stain to get some of that acid phosphatase and other materials into the swab, and then you add your reagent, which, like I said, contains a substrate in the chromogen, on the swab, and you get a purple color change. Okay. There's also, if you're dealing with a swab from a sexual assault kit, you need to remember that that's your entire evidence. So you don't just want to go squirting acid phosphatase on that swab, or you may be getting acid phosphatase all over your evidence that you need to use for later testing. You may need to send some to another lab so they can repeat your results, or do some evidence testing for the defense. So you don't want to use the whole swab. So normally if a swab is your evidence, you may take a little snip of the swab to test it for acid phosphatase, or you could take a piece of filter paper and moisten the filter paper and wrap that around the swab, and you're transferring any material from the swab, including semen and acid phosphatase, to the filter paper, and then you just add your chemicals to the filter paper. So it's sort of like swabbing the swab, except you're not swabbing the swab, you're just sort of sampling it with filter paper. All right. Okay. Either way, you sort of take a small amount and then test it. You can also test large areas. I should probably save this for the other class. You can also test large areas with the AP test. If you're dealing with a piece of bedding or a piece of clothing where you take the alternative light out and you see a whole lot of fluorescent stains, and some of them are faint, and you don't really see a strong indicator of where something's a semen stain, the other way to search a large area is by laying down large pieces of filter paper over the piece of clothing. So let's say that we have a pair of underwear here, trapezoidal underwear, I guess. You could lay like big pieces of filter paper over those. Let's say we got 2 big pieces of filter paper, and then you sort of draw with your pencil, mark where the underwear is, so and then you moisten all of this with water. Okay, so a little spray bottle of water. I have no idea what am I doing? That's a spray bottle of water. It looks like a hose or something. So you moisten it all with water and press the filter paper down on the clothing, and any place where there is a semen stain present, the acid phosphatase is going to be absorbed into the paper. You then take the paper down here off of the clothing, and you can spray the whole thing or soak the whole thing in acid phosphatase reagent. And then what will happen is wherever there was semen on the clothing, you'll get this little purple spot, and you know that that's where you want to target your further testing to see if you can find sperm cells or do DNA. Okay, so it's just another method for testing a large area. As we already said, other items are going to react with this AP spot test, specifically other materials with acid phosphatase, so urine, vaginal secretions, etc. So if your mock case is, you receive a piece of clothing with a suspected semen stain on it, you do the alternate light test, it fluoresces, you do the AP spot test, it turns purple. What are your conclusions? Okay, well, answer in the chat box. Okay, if you have a stain that fluoresces and tested positive with AP, does that mean it's definitely semen, it might be semen, or it is definitely not semen? Okay. Yeah, and Sarah comes in with right, and everybody's getting it right. It might be semen. Okay, so that's what now there may be two explanations for it, and if it's on the mock case quiz, the answer is going to be it might be semen because other materials may also contain acid phosphatase. All right. So if you have a weak reaction, the question is this a false positive? Is it a weak semen stain? And you just have to, again, use your experience as a forensic serologist to determine whether you want to move on with further testing or just stop right there. All right, like I said, it can be very important to go into court and say, yes, this is definitely semen. So there are two confirmatory tests for semen. Okay, the first, Christmas tree stain, is where we're going to be looking for sperm cells. The next, prostate-specific antigen, is going to be an antigen-antibody reaction that is similar to a pregnancy test. We mentioned this briefly when we talked about species testing with blood. Okay, let's talk about the Christmas tree stain first. It's not called the Christmas tree stain because it's festive or only done around the holidays or because Starbucks hates it. It's because you use a red stain and a green stain. Okay, and you have to remember, I mean, here, with the previous slides of sperm cells, they were nice and neat, and this is nice and neat where we just have sperm cells. But if you're trying to find sperm cells in a vaginal swab from a sexual assault kit, you're also going to have a lot of female epithelial cells. And if you don't stain the slide, you just get a slide with a bunch of like semi-clear splotches all over the place, and it can be difficult to find the sperm cells. Whereas if you stain the sperm cells, they're a little bit easier to pick out the red dots, and the tails are green, which don't really show up well in either of these pictures. But the process is where, or maybe that's on the next slide here. Yeah, the method is where you take a little portion of the stain or a little snip of a swab if it's from a sexual assault kit, and you put it on a microscope slide with a drop of water. You just put a drop of water right on top of it, and then you start to tease apart the fibers of the fabric or the swab. As you tease apart those fibers, then any sperm cells that are on that fabric will be released into the water. And, the sperm cells are dead at this point, so it's not like they're swimming around, but they're just released into the water. And then you apply your red stain and you dry it off, and then you apply your green stain and you dry it off. And after that, you can just slide that microscope slide under a microscope and literally look for sperm cells. Okay, at 1,000 times magnification, they are easy to see. Actually, we used 400 times magnification, and we could see them just fine. All right. Again, the big benefit of that. So if the mock case question is you do a Christmas tree stain test and you see sperm cells, what is your conclusion? Even though I know everybody hates to say it on a multiple choice quiz, the correct answer would be this is definitely semen. So it's okay to answer that question in this mock. It's not like true and false where if you see the word always, then it's always false. Although that's always never mind. But yeah, sometimes the answer is definitely yes or definitely no. Okay. You get a strong AP test, turns purple. You look for sperm cells, you don't see anything. So what's going on here? Either I got some weird that giving me a strong reaction, or maybe the person has a very low sperm count and I just can't find. Maybe they had a vasectomy. In that case, there is another option for a confirmatory test, and that is a test for prostate-specific antigen. Now, you may hear PSA talked about. Sometimes they look for levels in blood for people with prostate cancer, but the levels in blood are so low, and blood won't give a positive with acid phosphatase, that we can consider prostate-specific antigen as being specific to semen. So in other words, if we get a positive AP test and a positive PSA test, you can go into court and say, yes, this is definitely semen. Why? Because no other substance would give me a positive for this PSA test. Okay. So it's a protein produced by the prostate gland. Again, it has nothing to do with the amount of sperm cells or anything like that. And the test is an antigen-antibody reaction. Specifically, it is a card just like a pregnancy test. So you'll take a cutting of the swab or a cutting of the stain, throw it in a tube of water and shake it up. Any sperm cells that are in there, any PSA that's in there is going to go into the liquid form, and then you take a drop of that liquid, okay, and you apply it to the test well. Okay, just like when you put a bounty paper towel, the corner of a bounty paper towel in liquid, okay, that liquid is going to start to wick across and move across the T and the C area. Okay, embedded in where the T line is are antibodies to PSA. So if that lights up, it's a positive. Okay, and embedded in the C line is a control antigen, and it just is there to make sure that the liquid has made it all the way across the test. Okay. All right, there are three, we didn't go into this level of detail in the blood test, so we're going to go through it here. There are three potential results. All right. Now, notice the C line. So when I show you a picture of these and ask you, what is your conclusion based on this picture, you may see one of these three. The first thing you need to do is look at the C line, okay, and recognize that the C line is a control line. That just tells you if the test is working or not. Okay, so for the first picture here, yeah, this test is working. Okay, this test is working. Okay, no C line means it's not working. You can't draw a conclusion. It's inconclusive. If that happens, what do you do as a forensic scientist? You get another one out and try it again. If that one doesn't work, then you call the company and say, hey, this is the lot number. What's going on with these tests? None of them are working. All right, so that's inconclusive. Okay, but you just use it until you get the C line to show up, and then you look at the T line. If it's present, that means PSA is present. It's positive for semen. Okay, if there's not a line, then that means PSA is not present. It's negative for semen. All right, so that's a confirmatory test for semen. All right, remember, for semen, lights up with an alternative light, so that's how you search for it, with a UV light. We got one presumptive test, the AP spot test, and we got two confirmatory tests, Christmas tree stain or PSA. For the Christmas tree stain and PSA, if you see those on a mock case quiz, it's okay to answer definite yes. Unless the test is negative. All right, saliva. If you do a search for saliva on the internet, safe search on, although I guess it doesn't really matter with saliva, you get a lot of dentists and ladies getting saliva sucked out of their mouth by a dentist. You also get a Russian band called Saliva, who apparently, according to one student I've had in my over 10 years of teaching, is good, and I also think they're still around. And they're Russian. I'm not here to give a lecture about saliva, though. I'm here to talk about saliva. All right, let's think about blood. What does it look like? What's in it? Semen, what does it look like? What's in it? Saliva, it's clear. That's what it looks like. What's in it? Cells? Yeah, cheek cells fall out of your mouth into your saliva. It's not like there's specific saliva cells that are secreted into it, but cheek cells can be in saliva. What about enzymes? Probably. Tell us about the enzymes, Dr. Linville. All right, so our focus is going to be the enzymes. Other stuff is going to include cells. We have a lot of saliva each day. I'll spare you the details of talking about wrestlers and losing weight by spitting into a jar. Although for many years, students looked at me like I was nuts when I told that story, but then we had two wrestlers in class last semester, and they both confirmed that at times they have spit into a jar in order to lose weight before they wade in for a match. Anyhow, saliva aids in digestion. Okay, starts to break down your food. Most of us learned that in 3rd grade science or something like that. But the reason it breaks down food is there's an enzyme in your saliva that breaks down starches. Oh, does this sound familiar? Enzyme in a biological fluid. We're probably going to do some type of chemical test to detect that enzyme. Correct. Amylase is the enzyme that we're going to try to detect in saliva. Okay. It breaks down starch. It's high in saliva. It is found in other body fluids. So what does that mean? It means that this is a presumptive test. We're not going to get a definite yes here. Okay. But the way that we detect amylase is not through some complicated chemical reaction. Actually, all of these involve detecting whether the sample, the evidence sample, is breaking down starch. Okay, now we're not just going to rub a potato on the stain and see if it starts to dissolve, but there's 2 tests that we can do that involve starch. Okay, one is the starch iodine test. The other is something called Fadibus reagent that also involves starch. Okay, the starch iodine test, let's start with that. If you mix together starch and iodine, it turns a blue color. All right, some biology labs will have you like throw a potato in a tube of iodine and it turns blue, and then it proves there's starch in the potato or something dumb like that. But if you actually mix starch powder, iodine liquid, and gelatin, okay, or an agarose gel or something, you can actually form a gel that will turn blue because the gel contains both starch and iodine. Okay, this is more PowerPoint award-winning art. I don't know if I mentioned this before, but I created an award for PowerPoint art and I gave it to myself, so it's accurate when I say this is PowerPoint award-winning art. This is really my best work. What this represents is a petri dish that is filled with agarose gel that has starch and iodine mixed in it, so it turns a blue color. Okay, you take a little scalpel, you cut out the middle here, and then that is a hole in the gel. All right, the process for this is where you have your sample. Okay, let's say we cut out a little piece of a bandana that we think has saliva on it from our robber. Okay, you throw it in the tube, okay, with a little bit of water, shake it up. Any saliva that's on the fabric will dissolve into the water. Any amylase that is on the fabric will be into the water. Then you add a drop of that into the hole. Okay, what's going to happen? That starch is going to start to seep into the gel. If amylase is present, if saliva is present, then it will break down the starch and a clear color, the blue color, will disappear. Why? Because there's no more starch here. Can't react with iodine, can't return the blue color. If nothing happens, then that is negative. That indicates that saliva is not present. Okay? So it all involves, is starch breaking down or not? This test is a little bit unique because that blue color is disappearing. All right, Fadibus reagent is another way that you can test for saliva. It's sold as a blue pellet. It looks like a Flintstone vitamin, and it crushes up very easily. And at the small level, it's actually tiny starch balls, and they're covered with a blue dye. So what you're doing in this test is you're looking to see if those starch balls are broken down by the amylase and the blue dye is being released. Okay, so this is a little bit more complicated than some of the other tests, but we're looking to see if the starch is breaking down, if the blue dye is released. Okay, so this method is you take a small cutting of stain and you put it in a test tube. Okay, so here's our little piece of fabric that we want to know if it contains saliva. And you fill it up with a bunch of water, and then you put a crushed Fadibus tablet in here. So it's sort of floating around. The little starch balls with the blue dye are floating around, and it looks like a real chalky blue color. Okay, then you heat it up a little bit. Okay, you wait, give it some time for the starch or for the saliva to break down that starch, and then you centrifuge it. Okay, what does that mean? You centrifuge the tube so all of those starch balls as well as the fabric go down to the bottom. So what's going to happen is down here at the bottom, you're always going to have blue stuff. Why? Because it was attached to the starch balls and they get pushed to the bottom of the tube when you centrifuge it. What you're looking for is has any of that blue dye been released into the top? If saliva is present, amylase is present, the starch breaks down, the blue dye gets released into the top. Okay, so let's look at a picture. This is we did in class. There's always blue stuff down here on the fabric, so let's ignore that. What we're looking at is up here in this area. And we can see here, this is actually neat saliva on a piece of fabric. This is a 1 in 5 dilution, and then this gets more dilute, and then this is our negative control, no saliva. So you notice where saliva was present, a lot of blue dye has been released, and it's blue. Okay, and our negative control, where saliva was not present, no blue dye was released, and it's clear. So if you do this test, it's a quick way to determine, was there saliva on this fabric? Again, a presumptive test because other things will also cause that, will also contain a little bit of amylase and may cause a slight blue color. All right, searching. Whereas the AP spot test, that search with filter paper, isn't really done that often, search with Fatabis paper can be very useful. And let's think of this scenario, because why in the heck would you do this test anyway? We're going to get to this with the DNA slide, but like if you have this cup that I'm drinking out of and you want to sample it for DNA, you're going to swab the top. And you're not going to go back to the lab and say, gee, was there saliva on the cup? No, you're just going to try to get the DNA out of it. Because you don't care if I was touching or bleeding or whatever or drinking. You just care about, is there any DNA on there? Hopefully, some cheek cells on my saliva got transferred to the cup and I can do DNA analysis. But it's not like you're going to go into court and somebody's going to say, how do you know he was drinking out of that cup and not beep censored, you know, in court where they don't record it? I don't know. No, nobody's going to question it. It's going to say his DNA was on the cup. Everybody's going to go, oh, well, I guess that means he was drinking out of it, right? Okay. But a situation where you might care about locating saliva would be, imagine I rob a bank with a bandana around my face. So I'm saying, get on the floor, everyone. Please. I don't know, I'm a polite robber. Would you kindly give me some money, preferably in large bills? It's just ones get a little bit heavy and I've been having back issues. Not a very good bank robber. But as I'm leaving, I throw my bandana that I had around my face to conceal my identity. Okay, I throw it on the ground. Okay, you know that my mouth has made contact with that bandana in some location where the spit has been flying out of my mouth as I'm pleading and being polite. So it would be interesting to know, okay, where is the saliva on this bandana? You can't just extract DNA from the whole entire bandana. It's really difficult to do DNA testing on a large piece of fabric that has a little bit of DNA on it. Like even if it's a shirt that somebody wears, they probably have a bunch of sweat on it, but it's difficult to do DNA analysis because you can't just extract DNA from the entire t-shirt, or at least not yet. Okay. So what you want to do is locate the saliva stain. Okay, so how you would do that is you would unfold the bandana. Let's say it's a square one that you have folded into a triangle. And again, you would put large strips of filter paper, except this time it's not going to be filter paper. It's going to be called something called Fatabis paper. And what Fatabis paper is on one side it's just normal filter paper, and on the other side it has that Fatabis reagent, meaning all of those little starch balls with the blue dye attached sort of applied in a single layer. So it sort of looks like this chalky blue color on one side of the paper. You put the blue side down, you spray it with water, you add some pressure. In class we put a big glass a big glass plate on it and then add some weight to it. And you wait. If saliva is present on the bandana, it will make contact with that Fatabis paper. It will break down the starch. It will release the dye, and the dye will begin to leak through the paper. Okay? I know that can be tough to envision, but let's see what's going on here. What we have here, even though it's difficult to see, is there is a piece of circle cloth under here. okay, that I have applied a saliva stain to. Okay, and when the students received the cloth, it was divided into 4 sections. Okay, 1, 2, 3, or 4. Okay, they put a piece of Fadibus paper on top of that cloth. Okay, and then they sprayed it down with water and put this glass plate on top of it. Okay, you can see that on the fabric where the saliva was began breaking down the starch in the Fadibus paper, and the blue dye was released and seeped through the paper. So here it's very easy to say, okay, if we want to target this for DNA testing, we're going to target that area. All right. All right, what are the limitations to this Fadibus test? Well, it's a presumptive test. There are other body fluids with amylase in it, so there are possibilities for a false positive. Also, it's not very sensitive, so if you have a dilute saliva stain, if the bandana has been washed, or if it's a little bit old, it may not be as reactive, and the test may fail for older samples. Okay. All right. As I mentioned before, if we've done the Fadibus test and now we know where the saliva stain is, then we can target that smaller area for DNA testing. But unlike with blood and semen, it's very standard to the first thing you do is do the presumptive test to determine whether it's blood or semen. For saliva, if you get a swab from a cup, you're not going to do a test for saliva because, like I said, it doesn't really matter. The fact that it's saliva is implied. All right, so it's only going to be if there's a specific question whether this is saliva or not. Maybe you're dealing with a victim that claims they were bit, so you want to prove that the bruise wasn't just from being punched or hitting their arm on the counter 2 weeks ago, but it's from a bite mark. So you want to detect saliva in their clothing. Okay, that might be one reason for biting, or if you're trying to locate a saliva area. All right. Other than that, if it's a swab of a cup or a bottle or the mouthpiece of a cell phone, you're just going to swab it and go straight to DNA, not really bother with saliva. Okay, those are the main things that we do DNA testing for, and the main things that will come through a serology lab for testing. Now, we want to mention a couple other biological body fluids, I guess, since they're in the book. Okay, one is urine. Urine may be involved in cases of harassment, mischief, sexual assault. So you may have a urine sample that you not only care of whether it's urine or not, but you care who it came from. And really with urine, in those cases, you want to know who did it come from. Okay, and just like saliva, being able to go into court and say, yes, this was definitely urine, it may be of some use. Like if I'm claiming that students are urinating on my office doorknob, then I might want to be able to demonstrate that urine is present. But it tends to be very difficult to do a presumptive test for urine. Well, what does urine look like? It's clear to yellow. What is in urine? Are there cells in urine? Sort of. Some epithelial cells, but there's not that many of them. Not as many as in saliva or semen or blood, that's for sure. What about, are there enzymes or are there chemicals in saliva or in urine that we can do a presumptive test for? Yeah, there's urea and creatinine, but as it turns out, there's not a good color test for these. And it's possible that unlike saliva that has a lot of amylase or semen that has a lot of acid phosphatase, if you've been drinking a lot of water and your urine is dilute, then there's not going to be that much urea or creatinine in them. So it can be very difficult to do these tests, and the end result is forensic labs don't really do presumptive tests for urine. All right. So it's difficult to detect. The liquid nature spreads it out, so it's difficult to both do a presumptive test and to do DNA testing. Now, with DNA testing in urine, if you come and urinate all over my office door, then I'm not going to, it's spread out all over. I could swab all day. I'm probably not going to get enough DNA. But if you decide to harass me by urinating into a Dixie cup and leaving that in front of my office door, whoa, that would make me angry. But I could take that liquid urine sample, centrifuge it to collect the cells, and I could possibly get a DNA profile from a liquid urine sample. Now, it's rare that people harass other people by urinating into Dixie cups, but where this would be applicable is if we care about like a urine sample for drug testing, determining the authenticity of it. Well, I think this is Barry Bonds' grandmother and not his urine. You could do a DNA test on a urine sample because you could collect by centrifuging, collect the cells from it. If you have urine left in a toilet, at a homicide, it's possible, I don't know who would get this job, that you could scoop all of the toilet water and urine out of that toilet and then try to centrifuge and collect the cells. All right. But if they just urinated on the floor, then you're out of luck. Probably. All right, feces is sort of in the same ballpark as urine, where there's not really chemicals at a high enough level in there to do a test for. So urobilinogen is something that is in feces and not in that many other biological materials. But as the book says, if you need, like similar to urine that's spread out all over the place, if you want to do a test for urobilinogen, you need a good sample of feces. And usually if you have a good sample of feces, like not a poop dot, if you have a poop dot, then it's like, is that poop? I don't know. There's not enough for me to, determine if there's any urobilinogen in there. I need a bigger sample. All right. Whereas if it's a turd, then it's sort of like, is that a turd? And the guy next to you goes, that's a turd. And everybody's like, there's poop right here. You don't need to do the test. Okay. We don't need a urobilinogen card, nerd. I know a turd when I see it. Okay. Unlike the people at CBS who came up with their fall TV schedule. Am I right, everybody? I mean, I don't know what I'm talking about. I've watched too much television as a young person to where I feel like I should criticize television executives every so often. Anyhow, hey, at least they canceled Two Broke Girls. I apologize if you were a fan of Two Broke Girls, but in all honesty, there's something wrong with you. Okay, so it's where if you have a lot of duty, then you don't really need to do a test for duty. Okay. All right, what about, can you get DNA out of feces? It is possible. There are kits designed for extracting DNA out of feces. It is a challenge because there's not that many cells from the pooper in there. The person who deposited the feces, I guess would be the correct term there. There's a lot of bacteria, which can make it more difficult to get a DNA profile from the person or animal. But there are kits that are designed to deal with this high bacteria content and the other types of inhibitors that make recovering DNA difficult. These DNA kits for feces were not originally developed for human CSI investigations, because again, it's not like there's feces involved in a lot of investigations, but they were originally developed for helping to track animals, specifically like wildlife animals. And you can imagine if you want to know if a grizzly bear has been reported as being spotted in a camp region, and you're the ranger and you want to know whether it's a grizzly bear or just a brown bear, then you may come upon some bear feces and you might want to extract the DNA and have it tested for the species and determine that. As I always like to tell my students, hey, if you're going to catch Bigfoot, you're not going to get a photograph of him, walking around. You're going to find Bigfoot. If you want to track down Bigfoot, find Bigfoot feces. Okay, he has to do it in the woods. I doubt that he's bearing it. Well, I don't want to make any assumptions about how a mythical creature defecates in the woods, but look for the feces. Okay, collect it, send it in for DNA testing, and after they laugh at you, just write them a check. Hey, you can write me a check. I'll do DNA testing on it, and it'll come back that it in fact is probably not some type of man-ape hybrid. You want to test for chupacabras? You just do it. Find the feces. Let me say something about chupacabras real quickly. It's always a coyote with mange. Okay, it's never a chupacabra. All right. What about vomit? There's no test for vomit, but I'll show you this picture and then not ask any questions on the quiz or exam about it. All right, that's it. Hang out so I can get everybody's. Now, what am I going to do? I don't want to show you a black slide, but I don't want to show you that either. Hang out for a while so I can make sure I got you down for attendance. I know a couple of you that were here at the beginning. I have you down. Quentin, I don't think I have you down on the attendance list. Also, if you have any questions about the lecture or upcoming quizzes and exams, go ahead and ask them now. I'll keep a recording. It may be helpful to the other students in the class. Let's see here. Let's discard those. And then eventually I will stop the recording, and if you guys want to ask any other questions, you can do that as well. Go ahead and take attendance here. All right, John. Okay, and when I say your name, you can leave because I have you down for attendance. All right. Ooh, I do not have Brienne. Some of you I know have been here already before. All right, Brianna, you're free to go. Isabel, you're free to go. John Thomas, you're free to go. JR is free to go. Keith is free to go. Quentin, let me write your name in here. All right, so we have a question that I'm going to answer on. Okay, and Quentin, you're free to go too. We have a question in here that I'm going to answer on tape. Hey, get out of here. All right, does DNA break down differently in semen than it does in blood? That's a good question, Sarah. Really, what determines whether DNA breaks down is usually the surrounding environment, and more specifically, the bacteria and the nucleases. Like we talked about this a little bit when it came to packaging DNA. Now, if you just have like a dried semen stain on cotton versus dried blood on cotton, they're both probably going to break down very similarly. And that is, if they're dried out, there's not going to be a lot of bacteria growing in them, and the DNA is going to last for a very long time. So it really depends on where they're at. I mean, I can imagine if it's a semen sample that is sort of present in a female and it's been a while before the sample's collected, then maybe there's more bacteria and nucleases there breaking it down. The one advantage that semen has over blood when it comes to the DNA breaking down, or more specifically the DNA disappearing, is that the sperm cells are a lot tougher than blood. So the one scenario that we found in our lab is that in laundered stains, like blood cells, if you throw them in the laundry, they bust open, the DNA gets washed away very easily. Whereas with semen stains, sperm cells actually stay closed and they stay gripped to the fabric throughout the wash cycle. So you wash a blood stain, the DNA is gone. You wash a semen stain, the DNA can still be there after a wash. So that's really not the DNA breaking down, but it is, I would say, sperm cells are a little bit tougher than blood cells as far as protecting the DNA over a longer period of time. But for both, if they're dried out and nobody's messing with them, the DNA is going to last for a very long time. All right, so hopefully that helps answer the question. Okay, got to thank you there. All right, I'm going to go ahead and stop the recording at this point. And if you guys want to ask anything else, you can.